Hello, my name is BrooklynShy Charlea Leonard, and I am a senior here at East Central. And I partnered up with Dr. Leah Dudley, and we did some research with the McNair program and the NASA program and East Central University. And what we did was the variation in stomata and the roots of the three sisters. So what the three sisters are is they are a triad crop grown together, which are the corn, the bean, and the squash. And so it does have some historical significance. Native Americans in the past would use these triad and they would grow them together in their gardens because it would hold spiritual significance, and they believed that it was a gift from the Great Spirit. And so the legend has it is that these sisters work together, and so when you plant them all together, the corn tends to grow first, and it provides stability and support for the bean that grows next, and it tends to wrap around it, and it grows vertically that way. And then once the bean grows and it expands its leaves, it provides shade for the squash that doesn't need as much light as the corn and the beans do. And so that's how they work in unison and how they succeed and how they grow together. And that's why they're typically grown that way. And they're also grown in today's cultures and agriculture fields. And they are used in today's modern times. So what I wanted to do is I wanted to test two hypotheses. My first hypothesis was to see if stomata density had an effect on root weights. And so I wanted to see if the higher the stomata count in a plant led to healthier or bigger plants. And then for the second hypothesis, I wanted to see if the stomata density varied between species. So if there was a difference between corn, between bean, and between squash. And so how I went about that is we planted these seeds in mid-November. So we would take one seed from each species. So we would take a corn, a bean, and a squash, and we would put them all in the center of the pot, and then we had multiple pots, and these seeds were donated from the Chickasaw Nation and the Choctaw Nation. And so we would take these seeds, we would plant them all together, and then we put them in this light room, and the light that we had in there, they were under about 14 hours of light with this LED blue light, I believe it was. And they were kept at saturation, which means that they were watered constantly up until the point of mid-January, I believe, and that's when we started our dry down process and basically allowed them to die. And before that, I went in there and I looked at the leaves and I did epidermal casts on the ones that had appropriate vigor. And so what I would do with the epidermal cast is that I would take a leaf, so there was an upper side and a bottom side, and I would take clear nail polish and I would just do a couple strokes on each side, paint the bottom, paint the top, and then I would take pieces of Scotch tape and then I would set it on top and make sure I'd press down, make sure it stuck on there good, and then I would slowly peel it off, and what I would be left with is a cast of what was on the leaf, so I could see everything that was on the leaf at that time. And so I took those peels, I took those casts, and I put them on microscope slides, and then I put them under the microscope, you can see here. And then this was my field of view, so this was an example of a corn, the upper side of a corn. And so you can see here, you can see some of the stomata that are present. And how I counted these is I used one field of view. So right here, this is what I would use. And then I would count up and down and then left to right. And so that's how I got the stomata counts for the plants. And then for the roots, here you can see I'm holding some right here. I would just dump them out. So I would go into the light room and I would get some pots, and I would just flip them over and dump them out. And then I would carefully try to sift through them and try to separate them as carefully as I could to make sure not to have any error and break any of the roots. And so once I got to a point where they wouldn't really separate anymore, I would then dip them in some water, and that would assist in the separation process. And so then once I got them separated, I would pick them up and I laid them on the counter. And then once I would lay them out, I took a razor and I cut at the first lateral root of the plant. So then I would cut those plants and then I would pick them up and I put them in aluminum boats. And you can see here the aluminum boats with the roots in them, and this is the drying oven that I placed them in. And so they would stay in there for about 2 to 3 days to actually make sure that they were really dry before I weighed them. And so after they were dry, I took them to this really sensitive scale right here, and you can see here's an example of me weighing that one. And so then at that point, that was the conclusion of my data collection, and so I had already had the stomata casts, the stomatal density numbers, and the root weights. And so now it was time to put it all together and analyze it. So what we did, Dr. Dudley and I used R, and we put our data into R. So for the first hypothesis, we used an ANCOVA test. And here you can see this is all of the data. These are root weights versus stomata counts. So this was for the first one to see if the root weights depended on stomata density. And obviously looking at this chart, you can see there's no real positive line, there's no real negative line, they're just kind of scattered there. So the results were that there was no relationship between either of those, and that led to the rejection of my hypothesis for that one. And so for this second one, you can see here, we put it into R. We did an ANOVA test for this one, and you can see the means here. So this was the upper side of the leaf, and these were the lower side of the leaves. And then this orange here is the bean, the black is the corn, and the gray is the squash. And so what you can see here is that it does depend on the species. It does depend on the plant variety on how many stomata that they have present. And so that ultimately led to the acceptance of my hypothesis, where variety does play a role on stomata density. And so ultimately, to conclude this research, the hypothesis for my first one was rejected, suggesting that there may be some other ecological mechanisms for the three sisters, and that's how they succeed. But I did find one reference that showed there was a positive relationship between root weights and stomata density. And I think the difference with that one is that I had a relatively smaller sample size, where in that study they had a larger sample size. So I think that was one of the differences. But I did find that there was a positive relationship. And when you think about it, it's kind of almost like common sense. But with our research, we did not get that result. And then through the second hypothesis, to conclude that we did accept our hypothesis here because variety does play a role on stomata density. So it depends on the variety type of the plant, and that will determine how many stomata the plant has on the upper and lower sides of the leaves. And ultimately, our work led to the baseline knowledge of the three sisters and what we had gathered so far. And I would just like to hear my references that I used for this project. And then I would like to acknowledge East Central University, the McNair program, the NASA programs, because without them, I wouldn't have had the opportunity to do this amazing research and learn as much as I did. And then I would also like to thank Dr. Dudley and all of my co-researchers that I played a part in this project that I'd really like to thank. And I am thankful for this opportunity, and I am curious to see where this opportunity leads me and where this research takes us. But that is the conclusion of my presentation. Thank you, guys.